Hi, everybody. I'm Rivka Solomon from the Massachusetts ME/CFS and FM Association, also known as MassME. And I'm here to introduce to the larger ME/CFS community two wonderful researchers, Liisa, Selin, and Anna Gill of the University of Massachusetts Medical School. They're researching ME/CFS, Epstein-Barr virus, and multiple sclerosis. And their research shows some interesting new ideas on the possible cause of ME/CFS. And if that research bears out, then they might also have some possible blood tests that could be used to help diagnose the disease. That is a possible biomarker. In 2019, they got a Ramsey Award from the Solve ME/CFS initiative for researching altered T cells in ME/CFS. And we at MassME, the Massachusetts ME/CFS and FM Association, are doing all we can to support their efforts. So I would like to welcome Liisa, Selin, and Anna Gill to explain a little bit about their research. Please, Anna.
Hello, can we change the slide, please? So here on this picture, you can see me getting ready to process the blood collected from ME/CFS patients. First, I isolate one of many blood cells, which are called lymphocytes. I count and stain these cells with markers in order to see what features these lymphocytes have, and if there's any change in their phenotype and function. Next slide, please. Here I'm showing you in details how the blood processing is done. I work with collected blood under laminar flow hood to have my sample sterile and also to protect myself from any potential pathogens in the blood. The cells of my interest are collected after I separate them using density medium and then centrifuge. On this cartoon, you can see the layer of cells, lymphocytes, which I then run on a fax machine. Next slide, please. Here I'm showing you very quick peek how fax machine looks and works. Fax stands for fluorescence activated cell sorting. It has three main systems: fluidic to keep the cells moving into the right direction, optical to detect the markers on the cell surface, and also electronic to transfer this data into the plots you can see on the right hand side. This computer analysis let us focus on particular cells, how big they are, what markers they have on their surface, for example, the ones to tell us if they are naive or antigen experienced. And also we know their percentage, they function, if they produce any cytokines, chemokines to find different virus infection. Thank you.
Wonderful, Anna. Thank you so much. And thank you for working in the lab for us, for our community. Liisa, Selin, do you want to expand on some of what the research is that you're doing and some of your findings?
So what we've been finding is that it appears the major cause of ME/CSS is defects in your immune response. And the particular cells that we have been focusing on are CD8 T cells and a population of cells that express both CD4 and CD8, which very little is still known about, although this population was identified 25 years ago. The CD8 T cells are cytotoxic cells, and they kill virally infected cells or tumor cells, and usually function perfectly fine, but in certain circumstances, such as persistent viruses like hepatitis C virus, or in tumor environments, they become overactivated and exhaust. And in fact, we are finding that to be the case also in ME/CFS patients, although we have not yet identified what the possible antigen is, whether it is virus or autoantigen. We do find that there's a decreased frequency of these cells, and they have decreased function, decreased interferon gamma production, TNF, and cytotoxic activity demonstrated by CD107 A and B staining. They also express CTLA4, which is a marker of exhausted CD8 T cells. I should like to remind you that the state of T cell exhaustion is associated with increased secretion of interferon type 1 and TGF beta, as well as decreased cellular metabolism. And these are features that have been also described in ME/CFS. The CD4 positive, CD8 positive cells have functions of both CD4 and CD8 cells. They're also cytotoxic and make cytokines. But we find that they are increased in number, and this may be a compensation for the exhausted CD8s. We find ME/CFS patients have 8.5% in their peripheral blood of these cells compared to 1.2 in healthy donors. But not only are they increased, but they are making cytokines spontaneously. They're in vivo activated, and they differ between males and females. Females have a large population of Treg-like cells that are making IL-9, the cytokine that can activate mast cells, while the males have FOXP3 positive cells that are making IL-17, a pro-inflammatory cytokine. And these cytokines, as well as increased CD4 positive, CD8 positive cells, have previously been shown to be associated with autoimmune diseases. When we looked at their TCR repertoire, it strongly suggests that these are antigen-driven expansions. The clinical implications of these studies is that we have identified potential biomarkers such as a low CD8 frequency, altered CD4 CD8 ratios, high CD4 positive CD8 positive frequency, and functional studies can be done for CD8 T cell exhaustion. Furthermore, there are potential therapies that are still relatively experimental, but therapeutic clinical trials could be tried for ME/CFS patients. For instance, checkpoint inhibitors such as anti-PD-1 are being used to treat cancer and chronic viral infections. Anti-cytokine therapy such as anti-IL-7 is being developed and used to treat autoimmune diseases like inflammatory bowel disease. There's also this question that if CD8 T cells are exhausted, that might mean that ME/CFS patients have difficulty controlling many of the persistent pathogens that we coexist with, including viruses such as Epstein-Barr virus and CMV, and therefore antivirals might be help with the disease. Understanding the pathogenesis of ME/CFS, the IL-9 and IL-17 have receptors in the central nervous system, and thus they may contribute to the type of CNS disease seen in ME/CFS, such as brain fog and problems with the blood pressure. IL-9 is also a potent mast cell inducer and may contribute to allergies and mastocytosis seen in ME/CFS patients. I'll once again remind you that CD8 T cell exhaustion is associated with increased systemic levels of interferon type 1 and TGF-beta, and these cytokine abnormalities are associated with metabolic dysregulations that are very similar to those in ME/CFS. So potentially using TCR sequencing, we may be able to identify the major antigens, whether they're viral or autoantigen, that are driving or contributing to this disturbed immune response in ME/CFS patients.
Liisa, that is excellent. Thank you so much for going through those 2 slides so quickly and so efficiently and thoroughly at the same time. Could you please put it back to the first slide of the entire deck? I just want to say that these 2 wonderful researchers from Massachusetts, who again, we, the Massachusetts ME/CFS and FM Association, are supporting in any way we can, are of course, like all other researchers, searching for donors and funding so they can continue their excellent research. And we hope people might be interested in supporting that. And I think that's it. Thank you very much, Liisa. Thank you, Anna.
Thank you.
Thank you.